This video is going to describe the tryptophan operon, also called the Trp operon. It's another example of gene regulation in bacteria. So there's an additional video about the lactose operon. So this is going to start in a very similar way, but the example we're going to use is about the tryptophan operon. So again, gene regulation is this principle of turning off and turning on genes when it's needed in a specific cell. So we've seen this flow of information where DNA is used to make RNA copies in the process of transcription. And then that RNA can be used to make proteins in the process of translation. So while many proteins are needed to be present all the time in certain cells, there are certain proteins that are only needed at certain times. And so there's a process of turning off DNA, meaning it's not going to be transcribed, and so no RNA is going to be present. And then if there's no RNA present, then obviously proteins can't be made as well. And so this process of turning off a gene and then turning it on is called gene regulation. So in bacteria, they use a series of gene regulation called operons. And so we're going to be talking about two operons, the lactose operon in one video, and then in this video, we're going to talk about the tryptophan operon. So tryptophan is an amino acid. And if you remember, amino acids are the building blocks of proteins. So having all the amino acids present is really important because cells need to make proteins all the time to be successful and to survive. So having tryptophan present is really important. So what the tryptophan operon does is it encodes, so it's a piece of DNA that will ultimately make 5 different proteins that are these enzymes needed in the synthesis of tryptophan. So all 5 of these proteins need to be present to ultimately synthesize this important amino acid, tryptophan. And so since all of them are needed and they work as kind of a group together, they are regulated together as a single unit called an operon, more specifically the tryptophan or Trp operon. So again, operons are coordinated sets of genes that are regulated as a single unit. A helpful way of thinking about this is we often have multiple lights in our kitchen, but we just have a single light switch. So when you turn that one light on, you have multiple lights that are all on the same circuit and turn on and turn off together because they function in a certain task to light a certain area. So this is true in genes as well. If there is a certain function that multiple proteins play together and all are needed, then they're regulated or turned on together in one operon. And so that's true in this tryptophan operon as well. So operons can be either inducible or repressible, and this describes kind of how they are in their default state. So an inducible operon is off in its default state and needs something to happen for it to be turned on or to be induced. That's why I call it inducible. And then a repressible operon is the default state is it's on and something needs to happen for it to be turned off or to be repressed. So tryptophan is an example of a repressible operon, as we'll see in just a second. So if you've watched the lactose operon video, this looks exactly the same. These are regions of DNA with names of regulator, promoter, operator, and structural genes. And their definitions are exactly the same. So the regulator is a piece of DNA that makes an important protein called the repressor. And it can be far away from this other region. That's, again, what these dotted lines mean. And then the promoter is where RNA polymerase binds. So RNA is the enzyme responsible for transcription. And then the operator is where the repressor protein binds to turn off transcription. And then the structural genes are those genes that are responsible for whatever this operon is designed to do. So in the tryptophan operon, again, there are 5 proteins that are synthesized to make tryptophan. So these are the genes that are regulated in the operon. In tryptophan, in the Trp operon, there are, again, 5 structural genes that encode 5 proteins that synthesize the amino acid tryptophan. Remember what amino acids are used for. They're used to build proteins. And so when would it make sense in a cell to make the proteins that make tryptophan? So when tryptophan concentrations are already high, when there's an abundance of tryptophan, does it make sense to make protein proteins to make more tryptophan? Or would it make more sense to have tryptophan synthesis enzymes present when there's little to no tryptophan? So hopefully you've come to this conclusion that the Trp operon, energetically it makes sense for it to be on when tryptophan concentrations are low. So when there's little to no tryptophan, the cell needs to make tryptophan. That's what this operon is going to allow it to do. And then when tryptophan levels get high, that's a time where it makes sense to turn off the structural genes that make the enzymes to synthesize more tryptophan. So that's what we want to look at and understand. How is this operon going to achieve this end result of turning on when tryptophan is low and turning off when tryptophan is high? So again, the regulator is a piece of DNA that makes this repressor protein. Now, in this case, the repressor, well, it's just like the lactose, the repressor protein's always present. RNA polymerase is another protein that's constantly being translated, so it's these two proteins are present. But what's a little different about this one is that in the default state, the repressor is not able to bind to the operator. So this site is currently empty. So although the repressor is present, it is not active. It cannot bind to the operator in this state. So the repressor is not bound to the operator. And that, of course, allows RNA polymerase, shown here in green, to access their structural genes. So it's going to transcribe, and then those genes are going to be translated. So the operon is on. And another thing to note is in this diagram, there is no tryptophan present, or the concentrations are so low they're not shown. So when tryptophan concentrations are low, this Trp operon will be on. And so RNA polymerase can get access to that structural gene and transcribe it. And then that piece of mRNA is used to make 5 proteins. And these proteins are used to synthesize tryptophan. And so once all of these proteins are present, here's a tryptophan molecule. Of course, these enzymes continue to work. And so more tryptophan's made, more tryptophan's made. And this will continue over and over again until there is some abundance of tryptophan in the cell and it's now accumulated. Now, hopefully you've noticed this repressor has a binding site on it. And so when tryptophan has accumulated, tryptophan serves a role, it's called the co-repressor. So as we said at the beginning of this, the repressor was not able, it was inactive, it could not bind to the operator by itself. It needs something else, it needs the co-repressor. And so only when tryptophan becomes abundant in the cell, that's when it's able to, tryptophan is able to interact with the repressor, because it's the co-repressor. And then those two together, the repressor, co-repressor, those two together are now able to bind to the operator. So it's this repressor co-repressor complex that binds to the operator. And now this RNA polymerase can no longer transcribe the structural genes. So over time, as you might guess, the RNA will disappear. And then over time, these proteins will disappear as well. And then because the amino acids are used to build proteins, the tryptophan over time is going to be used to make proteins and will continue to decrease. And so you saw those disappear. And then if tryptophan continues to be utilized, this co-repressor will also be used. And so that site becomes empty again. And just as we saw at the beginning, when there's no co-repressor present or the tryptophan concentrations get low, then the repressor by itself can no longer bind to the operator, freeing up RNA polymerase to transcribe the structural genes, make the proteins, and then synthesize more tryptophan. So that gets us back kind of that original default state, making the mRNA and then making these proteins and then making tryptophan again. We can continue this in an endless loop when tryptophan accumulates so much so that it accumulates and binds to the repressor as the co-repressor, it then binds to the operator and the process continues. And so The operons are an important way of regulating certain genes within bacteria, and we group them into these two possible ways of regulation. So the trip operon is known as a repressible operon because something needs to happen to repress or turn off the gene, and that something is the accumulation of tryptophan as the co-repressor to bind to the co-repressor, binds to the repressor, and activates it.